Couple of quick announcements before we get started. If you don't have 18 other things going on this coming Saturday, a couple of really fun things should be going on. First of those is Ladies Lab Night, Portland State University. Basically, this is mostly to be for women, middle school, high school age, who are potentially interested in science, and should be a whole lot of fun. Multiple people, including from my lab, will be presenting on their work. That's from noon to 6:30, and Allison Campbell will be speaking at noon. She's the Associate Lab Director from PNNL, which is used to be where we built all the plutonium triggers for our nuclear bombs, but is now actually a really very, very world-renowned site for environmental research up in Hanford, Washington. So should be a lot of fun. And then more information about Ladies Lab Night here as well. That's one announcement. Just in case that's not necessarily your cup of tea, from 12:30 to 1:30 over at St. Mary's, just off of campus, Mario Capecci, who got the Nobel Prize in Medicine a few years ago for the development of knockout mice. And we're going to talk about how some of that works, and in fact how we're doing it slightly differently these days. He's going to be talking about the intersection of art and science. So again, that's from 12:30 to 1:30 over at St. Mary's. And then one more blatant plug. Speaking of announcements, there's this journal called Microbiology Resource Announcements, to the American Society of Microbiology. And if you look at the Board of Editors site, you may see a face that is somewhat recognizable to you. So otherwise, announcements-wise, I hope that I've figured out what the problem is with all of the recordings. Has to do with this funky little external USB thing. Of course, that's the first thing that comes to mind whenever you think about this. Just trying to use too much different technology here in class. And probably the easiest thing to do would be to just get rid of clicker questions, which some of you might like. So I don't know. I don't think it's such a good idea anyway. And that being said, some of you may have noticed that the recording for last time, the first one that I posted had a couple of holes in at the end. I filled those in and reposted that. There's still a section on regulation at the end, which I did not re-record. We'll be talking about that, sort of recovering that today. And there's also a link to last year's lecture that I put in the notes on D2L as far as that's concerned. So we talked about last time was, sorry.
I just had a question about last lecture.
Yeah, last lecture.
What do you mean by a leucine zipper interacts with an extended major groove?
Okay, leucine zipper interacts with an extended major groove. So it's not a leucine zipper. It's the basic leucine zipper, so a basic helix plus the leucine zipper. And the leucine zipper is dimerization. And you've got your basic helices that are hanging off on the end of it. Those interact with a major groove like this. And so it's a longer piece of major groove. It's not like the 3.4 nanometers distance, which you have with the two helix-turn helices binding in adjacent major grooves. So it's a major groove which is literally right next to where you have those basic helices. Yeah, other questions on the last time? And again, I'll continue to talk about negative regulation as far as that's concerned. So the whole idea of negative regulation, it's a little funky because you can also use negative regulation to turn on genes. And so the idea of negative regulation is you have a repressor. And that repressor will repress transcription from a particular gene. And that repressor can be bound to your gene, and usually, again, it's the promoter right next to it, in the absence of a ligand. And when you put in a ligand, then this repressor comes off. This is what happens with the lac operon. You put in lactose because all of these genes are going to be degrading lactose. On the other hand, you can have a repressor that binds in the presence of some kind of a ligand, like you have with the trip operon. So tryptophan is around. You don't want to be making all the genes that you need for tryptophan. In the absence of tryptophan, it comes off, and all of those genes can be made. So that's the story, and that's also to some extent what Monod and Jacob got their Nobel Prize for, was figuring out this whole idea of negative regulation. And I'll tell you a story in just a couple minutes about negative regulation, which is really kind of fun. But negative regulation, I think one of you, I think, asked me about positive regulation last time. Positive regulation is just the flip side. So instead of having a repressor, now you have an activator. And repressors block the interaction of RNA polymerase. An activator stimulates the interaction of RNA polymerase with a promoter. So here we have an activator protein bound next to the promoter. And that activator protein helps the RNA polymerase to bind to this particular promoter. And if you think about it, we talked about consensus promoters and non-consensus promoters, so sequences that RNA polymerases likes to bind to and sequences that RNA polymerases are not so good at binding to. Well, if you have a promoter that's not quite consensus, but you have an activator right next to it, then you can get the polymerase to come and bind there much more so than it otherwise would. Just like the case with negative regulation, activators also interact with ligands. And so just like we have with the negative regulator here, if you add a ligand, the activator falls off, so you don't have the polymerase bound. Here, if you have a ligand, it helps the activator to bind, which helps the polymerase to bind. And one of the examples of that is called this CAP or CRP protein. Two different groups of researchers. Some call it CAP, some call it CRP. It's exactly the same protein, either the catabolite activator protein or the CAMP response protein. So CAMP is the ligand that interacts with this protein, and it reacts to the presence of catabolites. So catabolites, these are those molecules that are getting broken down. And so presence of catabolites, you then need to make some of the enzymes that you need for this particular purpose. So great, we've got activators and we have repressors. Well, it turns out that some proteins can do exactly the same thing, just one protein. And so the classic example of this is one called the lambda repressor. So not surprisingly, it does serve as a repressor of transcription, but it also serves as an activator of transcription. So how the heck can you have one protein that serves as an activator and that same protein that serves as a repressor? It depends on exactly where this DNA binding site is. So if the DNA binding site is in the right place, so when this protein interacts with the RNA polymerase, it sits down at the -10 and -35, it acts as an activator. If, however, it's moved one base pair away, it still interacts with the RNA polymerase, but that RNA polymerase is not sitting on the right place on the DNA for the promoter, so it can't actually activate transcription. So it's literally the exact placement of where that binding site is for the activator or repressor that determines whether it acts as an activator or it acts as a repressor. And in the case of the lambda repressor, it literally is just one base pair. So it's in fact just one site that the lambda repressor binds to, and it represses transcription in one direction and activates transcription in the other direction. So it's literally where the binding is that is going to determine this. So with that, I wanted to revisit the lac operon. So last time we talked about the lac operon. Nice story. You have this repressor that binds right next to the promoter where the RNA polymerase is going to bind. In the absence of lactose, it binds. The presence of lactose, it comes off. Wonderful example of negative regulation. So repressor binding. So that brings me to my story, which is in this fun book called Operators and Promoters, The Story of Molecular Biology and Its Creators by Harrison Eccles. Hatch Eccles was one of my professors in graduate school. So I actually heard this story from him, and that's one of the neat things I think about molecular biology is a lot of these people are still around or very closely related. Yeah, we have the example of crazy Jim Watson, who's still around. But Hatch Eccles tells this story, and I'll read it here, where he says, a large international meeting on cellular regulatory mechanisms was held in 1961 Yeah, I wasn't quite born then. At the Cold Spring Harbor Research Laboratory, Jacob gave a triumphant presentation of the evidence for the operon model, which is all about this lac repressor model. And Monod concluded the meeting with a general discussion of regulation, including possible applications of the operon mechanism for developmental biology. Somewhere in between, I gave my first scientific talk. Hatch-Eccles, again, my professor from graduate school. To a large audience, a brief presentation about regulation of the enzyme alkaline phosphatase, which I've been studying with Alan Green and Anna Maria Torarini. I commented that control of alkaline phosphatase might be positive. The regulatory product might be an inducer required to turn on the phosphatase gene. I expected some mild interest in the generalization of the operon model from my heroes, Jacob and Monod. Instead, Monod ran to the front of the room, proclaiming, "No, we know that all regulation is negative. Moreover, we have mutants like yours, and we know how to interpret them." I slunk from the stage, and Garen and I retired to the bar to heal our wounds. Subsequent experiments demonstrated that phosphatase, as well as many other biological regulatory systems, exhibit positive regulation. Ironically, even the lactose operon, which is why Jacob and Monod got their Nobel Prize, and lambda use dual control, positive and negative. So that's the rest of this slide, which is the revisited lac operon. The lac operon only is properly regulated with both negative and positive regulation. And the positive regulation comes from the cap protein that binds to cyclic AMP in the absence of glucose. And it turns out that all of Jacob and Monod's experiments were done in a minimal media that didn't have glucose in it. So this was on all the time, and they never noticed. But here, this is really nice because basically what it does is it integrates two different signals. So we talked about lactose, right? You don't want to make the genes that are going to break down lactose if you don't have lactose around. So that's the reason you have the repressor. On the other hand, you don't need to make these genes if you have glucose already. So what lactose is a disaccharide that breaks down into glucose and galactose. If you've got glucose around, there's no reason to break down the lactose in the first place. So it's two different signals that are regulating this one set of genes. So you only really have good expression in the absence of glucose. So you have this activator protein that's bound to cyclic AMP. Cyclic AMP is a signal which says you have low glucose inside the cell. That's the catabolite activator protein. That then binds to DNA right next to the promoter, helps the polymerase bind, but it will only work in the absence of the repressor protein. So repressor protein in the absence of lactose, bound here because these genes can't do anything if they don't have any lactose around. So the absence of the repressor, the presence of activator, now you have full expression of the lac operon. And so this whole idea, the concept of having two different signals regulating one promoter, is something that is really general. And we'll talk about it for the next couple of lectures in terms of what actually is going on, particularly for eukaryotic regulation systems. But I think it's really kind of, well, amusing is maybe not exactly the right term, that these two guys get the Nobel Prize for figuring something out that actually they were wrong about. So that's not the only kind of regulation that we have in bacteria. This is just probably the best studied of those. Again, positive regulation, negative regulation, etc. To get activation, this is now in bacterial promoters, you have your activator and your repressor, but you also have the RNA polymerase. And the RNA polymerase has the sigma factor that's associated with it. So that sigma factor that we've talked about so far is the sigma 70, at least in E. coli, 70,000 molecular weight. When on an SDS page, that's the size of it. That sigma factor, together with the polymerase, means that the RNA polymerase binds to -10 and -35. And those consensus sequences that we already talked about that I can't remember, so I don't expect you to remember their sequences either. On the other hand, it turns out that the sigma factor can be exchanged. So you can take a bacterial RNA polymerase and change the sigma factor that's associated with it. And that's, again, it's just a protein. But those sigma factors now have different binding specificities. So instead of binding to the -10 and -35 consensus sequences, a polymerase with, say, the sigma 32 sigma factor binds to very different sequences and different consensus sequences. So in the presence of a particular sigma factor, and it turns out this sigma factor is induced by a heat shock, and one of the things that happens is when you have a heat shock, this gene is turned on. It then activates genes for a bunch of chaperones. Chaperones important for protein folding, also a lot of the heat shock proteins. So this is how those genes are being regulated, how they're being turned up in the presence of a heat shock, is just by expression of this secondary sigma factor. There are lots of different sigma factors, at least 6 sigma factors present in E. coli. Some other bacteria have even more of these. And this has been taken advantage of by a number of viruses that have this, I think, really fascinating. Again, I'm virocentric. So what happens here is you have genes in the viral genome that encode different sigma factors. So this is the gene that comes in the viral genome. When the virus first infects a cell, the cellular RNA polymerase will transcribe these genes, including this sigma factor gene. This sigma factor gene now will reprogram the RNA polymerase to bind to some viral genes that otherwise this polymerase would never bind to. Some of those are actually yet another set of viral sigma factors, which will reprogram the polymerase to bind to yet other genes. And this is a really nice way of getting temporal regulation. So over time, the first sets of genes are made, then a second set of genes are made, and a third set of genes are made, because it takes time to transcribe and translate all the proteins that you need to reprogram the polymerase. So a really nice way of getting temporal regulation is expressing these different regulatory subunits, and literally specificity subunits of the RNA polymerase, over time. So it's a different way of doing regulation. We've got activators and repressors binding right next to the promoter, or we can play around with the RNA polymerase and that RNA polymerase specificity. There's another kind of bacterial gene regulation, which also uses activators. But now these activators are at a very strange place relative to where the RNA polymerase binds to the DNA. These are DNA binding sites for activator proteins that then somehow act on an RNA polymerase that can be literally thousands of base pairs away. Now, that's not how activators and repressors are supposed to work. Again, just like Jacob and Monod were saying, they're literally physically blocking the polymerase from binding or physically helping the polymerase to bind to a particular position where it's going to be the promoter so it can transcribe these genes. So this case called, oh, pardon me, we should get our clicker question out from in front of Sydney's face, is, again, just like, in fact, these original gene regulation proteins were found in the lag operon, was genetics. And you make mutants, you have mutations in specific genes, you see that there are problems. So, for instance, you make a mutation in an operator sequence that a repressor would bind to. The repressor doesn't bind there anymore, so you have lots of gene expression. So in this case, there were mutations that were made in a particular sequence of DNA that was a long way away from the promoter, but still blocked any kind of activation from going on. So to make a relatively long story short, and that what's happening is you have binding by an activator protein to a sequence, and again, this can literally be a sequence thousands of nucleotides away, that interacts with the polymerase by forming this looped structure. And this looped structure right here, you can actually see in an electron microscope. So this is a complex of activator proteins. This is the RNA polymerase. And in the electron microscope, you can see the DNA in between them actually looping out. This is from Wen Su in Sidney Kustu's lab at UC Berkeley about 10 years before I joined Sidney's lab and did my PhD working with her. Most brilliant scientist I have ever met. Unfortunately, died a couple of years ago. And these are Winogradsky columns in the background here that she had in her office. You taking microbiology, you'll learn all about Winogradsky columns. Great fun here. So this looping interaction loops to the polymerase. But you should go, well, wait a minute. Don't activators bring polymerases to the promoter? What's this polymerase doing sitting at the promoter? So this polymerase is sitting at the promoter because it has one of those different sigma factors that's associated with it. This particular one is sigma 54. That number is not important. But what this sigma factor does is it, just like all the other sigma factors, gets the polymerase to bind to a different sequence. But unlike the other sigma factors, this polymerase will not make an open complex. So it doesn't pull the two strands of DNA apart until it has this interaction with this protein bound a long way away. And there has to be ATPase activity. There's ATPase activity by this activator protein. And then that causes open complex to form, and then you get transcription taking place. So this whole process of the protein bound a distance away from the polymerase that it's acting on, this is what's called an enhancer. And these enhancers are DNA sequences that regulatory proteins bind to, and they're far away from the actual promoter where you have the polymerase which is going to do its job. I spent about 6 years of my life showing that this interaction here is a very transient one, but we won't get into any more details about that. But I did want to talk a little bit more about these enhancers, because enhancers, as it turns out, are the way that almost all eukaryotic gene regulation takes place. It's the activity of these enhancers. So enhancers can act literally tens of thousands of base pairs away from a promoter. They can also be downstream of the promoter rather than upstream of the promoter, and they can even be on different DNA molecules. And that was one of the other proofs that somebody else in Sidney's lab showed at about the same time. One of the issues that people had were, does this activator actually bind to the DNA and then scoot along the DNA until it gets to the promoter and then interact directly with the polymerase? Or is it literally looping through space? And the way that they did that was they took catenanes. And we talked a little bit about catenanes before. You need topoisomerases after you've done replication because the E. coli genome is two circles that are linked to each other. Well, if you put a enhancer site on this circle and a promoter on this circle, if they're separate from each other, this guy can't activate this guy. But if they're connected to each other, this catenane interaction, then they can activate each other. And that literally says this has to be through space. It can't be zooming along the DNA. So this is, I think, pretty good evidence that we're getting DNA looping. Again, the electron micrographs, but also these catenane experiments. Now, it turns out you can play around with this. You can move these enhancers, and this is exactly what the Kustu lab did, further away from the promoter or closer to the promoter. And it turns out there's actually an optimal distance, which is about 500 nucleotides away from the promoter. Now, why is that? So that's because DNA is actually not that flexible. If you think about it, this structure is not terribly flexible. And anytime you're closer than, again, about a couple of hundred nucleotides, it's actually hard to loop the DNA around. And so this is just what you see in terms of all of these activators. So basically, right next to the promoter is great because you have a direct protein-protein interaction. About 100 nucleotides away is bad because then you can't bend the DNA in between the two. But then as soon as you get to 100 nucleotides away or further, then you can have this looping interaction that will give you transcriptional activation. So that's bacterial transcriptional regulation. Any more questions for me before I ask you one? Yes, in the back.
The tryptophan regulatory mechanism, is that kind of a special case? Is that also considered a negative regulation?
So the tryptophan operon. So that is not a special case at all. It's a very general case. So that's negative regulation because there's a repressor that binds to the DNA. And that's actually what Jacob Monod thought was happening in Hatch-Eccles' case, is that they had a binding of a repressor that was then removed when you didn't have tryptophan around. But repressors can either bind in the presence of ligand or the absence of ligand. Okay, other questions? Okay, let's see if we can get this to work now. The lactose operon is subject to which of the following? Negative regulation, sorry, transcription regulation, positive transcription regulation, regulation by DNA looping, both negative and positive regulation, regulation by alternative sigma factors. Ten, 5. We're still aiming for 100%, right? Are we there? Oh, so close. So yes, they're both negative and positive regulation, and Jacob Monod were wrong at least the first time around. Okay, so now let's shift gears and talk about eukaryotic transcriptional regulation. This is basically, hopefully anyway, a review. We've mostly spent time talking about this whole constellation of proteins right here. We've got our RNA polymerase. We've got all of the general transcription factors next to a TATA box, downstream promoter element, etc. So this is where you have the RNA polymerase actually binding. But, as I kind of mentioned already, a lot of the interactions that are happening are through enhancers, and as we'll see later, also silencers, which are DNA binding sequences which can be long distances away from the promoter. And when I say the promoter here, also people talk about the core promoter, or literally it's where the RNA polymerase is going to bind and start transcription. So this can be literally millions of base pairs in length, all of the regulatory sequences which are going to regulate, actually in one case, one particular gene. On average, it's actually about 50,000 base pairs that are involved in regulation of any particular promoter, which is still pretty amazing to me that you've got all this DNA which is just involved in one particular promoter. Now, one thing to remember here is that we've only got about 20-odd thousand protein-coding genes in our genome. And we've got 6 billion base pairs, although there are 2 copies of them, so it's better to talk about 3. So there's a lot of extra DNA, or quote-unquote junk DNA, but if that junk DNA is actually involved in binding to gene regulatory proteins, then maybe that DNA is not junk after all. So lots and lots of pieces of DNA. These regulatory proteins, they still have to interact through protein-protein interactions with everything that's present at the core promoter. So sometimes these interact directly with some of these general transcription factors, and that's what's supposed to be shown down here. But very often they interact with this big purple thing here in the middle called the mediator complex. And the mediator complex is 20 to 30 proteins. It depends on what organism you're looking at, and literally just seems to serve as glue between all of these regulatory proteins and the RNA polymerase itself, and basically determining whether you're going to get transcription from that particular gene at that particular time or not get transcription from that particular gene at that particular time. So lots of people have been working on this whole process and trying to figure out what proteins are interacting in what way with what parts of the polymerase. As I mentioned before, between 5 to 10% of the genes in the human genome, and most genomes actually, are these regulatory proteins, these gene regulatory proteins, which are going to interact with DNA in some way. And again, we talked about some of those ways that you detect that last time. But they're very small proportions of proteins. 0.1% of actual proteins are these gene regulatory proteins. So actually working with these proteins and purifying these proteins is really drag and has been very complicated and difficult to work with. So why should we care about this? As I mentioned, most of the eukaryotic genes are regulated this way. But it turns out that expression of just 4 of these gene regulatory proteins are sufficient to get fibroblasts to become pluripotent stem cells. So what the heck is a pluripotent stem cell? Can't even say it. So pluripotent stem cells are basically cells that you can take and they can develop into basically any different cell type. And so they used to be people had to get embryonic stem cells, ES cells, which were really hard to get. You had to get them from embryos. And then that was the only way you could do gene modification. That's exactly what Mario Capecci and his colleagues did to make knockout mice. Get embryonic stem cells, modify those embryonic stem cells, and then use those to develop an organism. Well, these pluripotent stem cells, and more importantly, the induced pluripotent stem cells, you can basically do everything that you would otherwise need embryos with. You can just use fibroblasts to do this. And fibroblasts are skin cells, so relatively straightforward. And so they, the group here that worked on this, and I've forgotten their names, it was a Japanese group, basically showed that expression of 4 of these transcriptional regulators were sufficient to get these fibroblasts, to become pluripotent stem cells. And they made this discovery in a big paper that they published in 2009. Literally 3 years later, they get the Nobel Prize. I think it's one of the records between actual publication of some of the data and getting the Nobel Prize. And it turns out that in some cases, you can actually only express 1 of these proteins and get fibroblasts to convert at least into something that looks like a pluripotent stem cell. And I just give you the link here. We're not going to look at that. One of the ways that this was done, which I think is a really fascinating process, used something called CRISPR-A, which is activation based on CRISPRs. And I hope that my graduate student who will be giving the lecture on CRISPRs a week from today will also talk about some of this CRISPR-A. It's a really neat mechanism. And it turns out that that particular activator actually changes chromatin structure much more than turning on an activator per se. And we'll talk more about that a little bit later on. So this is just the cartoon way of looking at this. Fibroblast cells, again, these are derived usually from skin or some other kind of epithelial cell. Why epithelial cells? That's because they grow, and they're growing constantly, so they've got active telomeres, etc. And then just by treating with a subset of proteins, in this case there are actually 6 of them, but KLF4, SOX2, CMYC, and OCT4. Nanog, this protein that we talked about before in terms of binding to a particular binding site and then interacting as a heterodimer, we talked about last time, and then another protein called Lin28. These then, if you put them into cells, express those proteins, I make lots of those proteins, those will then become these induced pluripotent stem cells that then can develop into cardiomyocytes, which are heart muscle cells, adipocytes, flat cells, neurons of very different kinds, immune cells, etc. So a really incredibly powerful technique to get these kinds of cells. And one of the great things about it, just a second, is that you can start here literally with the cells from one particular patient, and now they're genetically matched to that patient that you started with in the first place. So a lot of personalized medicine is based on being able to take some of these cells from the patient, modify those cells, and put them back into the patient. Yeah?
Did you want us to know those proteins?
No, these proteins, the CMYC and NANOG and all of these other ones, I would say that, you know, KLF4, SOX2, OCT4, and CMYC are pretty good in terms of things to think about. We're going to talk more about them later on. So as far as the exam is concerned, that there are 4 that they got the Nobel Prize for, I think it's a reasonable number to remember, and that maybe you can get away with 1, I think is also good. Which that one is, actually there are 2 different ones that you seem to be able to get away with, so I won't be testing you on those. Okay, more questions. What's going to be on the final? I don't know, I haven't written it yet. Okay, so one question that I always ask is, so how much of the genome is involved in these different binding sites, and how are they being regulated, etc. So fortunately, there's this whole project which has looked at exactly this question called the ENCODE project. I just wanted to take a quick look at this. So this is also called the, it's the Encyclopedia of DNA Elements. And when they say Encyclopedia of DNA Elements, basically what people have done is they have gone into the human genome, and actually the mouse genome as well, and just tried to find where all the promoters are, all the binding sites for transcriptional regulators, all of the genes, all of the exons, all of the introns, all the places where you have modified nucleosomes, etc. So that, fortunately, somebody else has done. And so you can go to this website, the ENCODE website, and look up all of these experiments that other people have done, and then look for your favorite gene or something that you happen to think is important for cancer development or something like that. So this is people have done all of these really high throughput screens, and in fact what we talked about last lecture with the chromatin immunoprecipitation of trimethylated lysine 27 in histone H3, that was contributed to some of these experiments, because now you can go in and say, okay, I'm interested in this gene, I've got this part of the sequence, it turns out that it's highly associated with histone H3 trimethylated under these particular conditions. So this is just a really wonderful resource for looking at a lot of these different elements. And at the last count, it was over 1.3 million elements. And these are then probably regulatory elements that are present in the genome. And then just again for comparison purposes, we've got about 20,000 protein coding genes. So you can do some of the math, but it means that you have literally tens of different regulatory sequence or regulatory elements that are going to be interacting with any given single gene. And what that means, of course, is it's rather complicated and confusing trying to figure out exactly what's going on with any given gene. So there's still lots of work for all of us to do for a long time. So that's big picture. Now let's focus in and look at a few gene regulatory proteins that are actually pretty well understood. And these are actually vastly oversimplified systems, probably not unlike the case where we have the lac operon and Jacob Monod thought it was just negatively regulated. So here we're looking at now one of these gene activator proteins, also known as an enhancer binding protein. So just as a reminder, the enhancer is the sequence right here, the yellow, which, sorry, the green, I'm not that colorblind, green on the DNA, and then the enhancer binding protein that associates with it. Almost all of these enhancer binding protein, gene regulatory proteins have multiple domains that they're made up of. And at a minimum, the enhancer binding proteins are going to have 2 domains. They have a DNA binding domain and an activation domain. These activation domains, people tried really hard to say, what's the specificity of this activation domain? And they looked at lots of different proteins and found out that these activation domains are basically sticky. Okay, great. How useful is that? But it makes sense because that's now what's going to be interacting with the other proteins, particularly the mediator complex and then other parts of the general transcription factors, in order to get the polymerase to the promoter to make sure that it transcribes the genes. There are lots of different ones. There are acidic ones. There are proline-rich ones. There are glutamine-rich ones. Each of these activation domains don't seem to have any like really nice rule to them. So how did people figure out what each of these domains were actually activation domains? One of the ways that they did that was in this kind of experiment right here. So in yeast, Saccharomyces cerevisiae, there's a gene for galactokinase. And it turns out galactokinase is a really easy gene to study, but they wanted to study, in these particular cases, the activity of a gene that otherwise was not in yeast at all. And it turns out that a really good gene to use for that is the LAC-Z gene, which is a beta-galactosidase which breaks down lactose. And in fact, that's one of the genes that's in the lac operon in E. coli that Jacob and Monod were studying. But it's really nice because it's an easy gene to study the activity of. So the gene is transcribed into RNA. That RNA is translated into protein. That protein has enzymatic activity where it takes lactose and breaks it down to glucose and galactose. That's nice if you've got a good way of detecting glucose and galactose, but what's usually happening is people will take a dye, which is colorless when it's not cleaved, and then goes blue as soon as it's been cut. And we'll see a number of examples of this a little bit later on. And you can put that dye on the yeast cells, and if the yeast turn blue, that means that this beta-galactosidase gene has been turned on. If they do not turn blue, that means that gene is off. And so they put this gene into the yeast and then started to play around with the gene regulatory sequences and the gene regulatory proteins that were next to this gene, which is being controlled by this promoter, typical Tata box. So what they found is if they just put this reporter gene next to the normal binding site for this Gal4 protein and the promoter for that and a normal Gal4 protein, this gene was expressed, the yeast became blue. If, however, they take the Gal4 enhancer binding protein and put a DNA binding domain on it that doesn't match this sequence, then you don't get the yeast turning blue. It's not turned on. But if they now switch out the DNA sequence to the DNA sequence that now interacts with this protein, they now get activation. And what that says is that the activation domain is completely independent from the DNA binding domain. So you can mix and match activation domains and DNA binding domains, and what ends up working is where you have a DNA binding domain that interacts with that specific DNA. And also, people call this a modular system, basically because you can mix and match. You can mix and match activation domains and DNA binding domains, and they really do seem to be very independent of each other. So what do these activation domains do? Well, they're sticky. Well, what do they do because they are sticky? It turns out that there are lots of different things that they can do. Probably the easiest way to think about how some of these activator proteins do their jobs is they actually will interact with the general transcription factors, TF2B, TF2A, very much TF2D. And interaction of TF2D of these activator proteins, so you have the activator protein that binds to DNA, and then it helps these general transcription factors bind to a promoter. Once these general transcription factors have bound to the promoter, then you can have the rest of the polymerase that interacts with the promoter. And once the polymerases interact with the promoter, then you have activity of TF2H, helicase activity, etc. Then you'll start to get transcription that takes place. As I've mentioned before, very often the mediator complex is between this activation domain and the RNA polymerase, which brings everything together. And one thing here I wanted to mention is this term called the holoenzyme. The holoenzyme is the RNA polymerase plus a bunch, if not almost all, of the general transcription factors. So people argue, and it's actually probably different for different genes, whether you get first you have binding by TF2D, then you have binding by TF2B, then you have TF2E, then you have TF2F, then you have binding by the polymerase. In some cases, it seems that the polymerase comes in with this whole bunch of other proteins it's already associated with, and then will associate with the DNA. And that's the process of the holoenzyme. But it does seem that you have two different kinds of interactions and probably present at different promoters. In some, you need to get those general transcription factors bound first, and in other cases, you can bring in the whole holoenzyme itself. And remember, the holoenzyme is pretty darn big relative to the DNA, and particularly the DNA when it's bound in chromatin, which we'll get to in just a second here. So one of the things that you may have noticed is when we looked at activation at the whole promoter, this is a direct interaction. So DNA binding protein activation domain interacting with mediator or with these general transcription factors. Turns out that there are also lots of proteins which act as co-activators, or I should say co-regulators. So these are proteins that are transcriptional regulators. They can activate transcription, they can suppress transcription or block transcription. But what's really weird, and when people were looking at these, they found they didn't bind to DNA. So how could you have a gene regulatory protein? What gene regulatory proteins do is they bring the polymerase to the promoter. How can you have one that's not interacting with DNA that will still help bring the polymerase to a promoter? And the answer to that is that they actually then interact with DNA binding proteins. So these are the co-activators and co-repressors, which bind to proteins through protein-protein interactions. And then this co-activator protein that's associated with the DNA only through other proteins can activate transcription. And it turns out you've got lots of different activators of transcription, co-activators, co-repressors. Sometimes they're using the same DNA binding protein, like this green DNA binding protein here, can interact with a co-repressor, can also interact with a co-activator, turning genes on, turning genes off. And again, a lot of this is context dependent. It's going to depend on exactly which of these transcription regulators you have in a cell at any given time. Again, this is why it's still pretty complex and complicated, and why we still have lots of work to do. And all of us will still have work to do. So I wanted to show just a couple of different ways of looking at this in a context now of chromatin-bound DNA. This is just different ways of looking at this. Other than the textbook, it's still basically exactly the same idea. The RNA polymerase needs to interact with the core promoter, which contains the TF2B recognition element, TADA box, initiator element, downstream promoter element. Again, this is about 100 nucleotides in the core promoter. How that gets regulated is by gene regulatory proteins that are either right next to the RNA polymerase and the general transcription factors that interact directly. These can also interact through co-regulators. You also have enhancer proteins that interact directly, this bottom is mine, or through co-regulators. And this can be upstream or downstream, relatively speaking. So now my pointer died, which is, oh, well, maybe not. We'll see. I can't move anything around, but I can advance with it. OK, so I'll start to use the other pointer here. So how do we get this process to take place? Most of the genes in your genome have nucleosomes associated with them. This complex here, which actually isn't even drawn to scale, is far too big to associate with the DNA that's associated with nucleosomes. Maybe a little bit of nucleosome breathing. You can have binding of one or two proteins, but you're not going to get the whole holoenzyme, not the whole pre-initiation complex associated with the DNA at one particular time. So usually the first thing that happens is you have a gene regulatory protein that's going to bind specifically to the DNA. Once it's bound, then it helps to allow other proteins to bind. It may be, and in fact in many cases is, directly through the activity of some of these co-activators, changes the structure of chromatin right here, which then allows the RNA polymerase to bind. Once you have RNA polymerase binding, then you have helicase activity, you have transcription that takes place, modifications happen on the C-terminal domain of this RNA polymerase. You also have a number of proteins that are associated with the polymerase moving along as it goes through the DNA. Gets to a termination point. That termination, everything falls off. You go back to this complex here, and the whole process goes around and around. So Usually that first step is some kind of gene regulatory protein. And again, in terms of here, we're talking about activation of transcription that comes in and binds to the DNA. And it's that first step that leads to all of the other processes. So what's happening when you have the binding of this gene regulatory protein? In many cases, these gene regulatory proteins actually will, can we get things to move here at all? No. Maybe, maybe not. There we go. Some of these proteins, in addition to binding to the DNA and interacting directly with the polymerase, they also have structural roles. And this makes a lot of sense if you think about the fact that many of these enhancers are a long way away from the promoter. So we'll just take here down at the bottom, this is your promoter sequence right here. Here's where you have to have your RNA polymerase that associates with the promoter. You're going to give yourself transcription. On the other hand, you need to have these gene regulatory proteins that are bound a long way away that interact then with the promoter. Turns out you can stimulate the interactions of these DNA binding proteins with the promoter if you have a DNA bending protein somewhere in between. So instead of just the DNA flopping around at random and then associating with everything in the pre-initiation complex, now if you have bending, that will help those other proteins nearby to bind with higher frequency, and the higher frequency they're bound, the higher frequency you're going to end up getting activation of transcription. Another thing that this DNA bending does is it gets rid of nucleosomes, because nucleosomes can't bind to this heavily bent DNA. Once you get rid of nucleosomes, then you can have association of all of your proteins at the promoter. So that's just binding. We've got activators interacting. But what do you see with the histones as you have transcription actually taking place? And this is just a cartoon of one particular promoter. And actually, it should be a gene. Transcription start site, that's the +1. We've already assembled our pre-initiation complex right here. Then the RNA polymerase starts to transcribe. And it turns out that part of the proteins that are bound to the C-terminal domain of the RNA polymerase II are proteins that are histone methylases. And so these histone methylases actually start to methylate histones that reassociate with the DNA after the polymerase has gone through it. And basically what this does is it says, OK, this gene is just being transcribed. We don't need to transcribe it again. And usually it's just that one round of transcription or few rounds of transcription. And after a gene has been transcribed, then it's silenced for more kinds of transcription that takes place after that. And that's just shown here by these little purple flags. These are now all methyl groups that have gotten added to histone H3 in this process. So this is transcription. How do we have these interactions of gene regulatory proteins literally tens of thousands of nucleotides away from a particular promoter? How do those, totally over-anthropomorphize here, enhancer binding proteins know that they need to interact with this promoter and not that promoter over there? Because they can interact through space. At long distances, they're away from each other. Well, it turns out that there's these processes called insulator elements. Now, what's an insulator element? An insulator element literally insulates the region of DNA that has your promoter and your gene regulatory proteins attached to it from all of the other ones. Now, how do insulators work? Insulators are literally sequences in your DNA that are going to interact with proteins and your insulator binding protein. And that insulator binding protein will now take this piece of DNA, usually as a looped structure. And so these loops should sound really familiar, hopefully, because we've talked about them before. Chromatin structure, these loops are where you're going to have expressed genes, but also where you have these regulatory sequences. and those regulatory sequences, which are now just going to work on the genes that are next to them, and next to them can be literally tens of thousands of nucleotides away, but not interact with genes that are on a different side of an insulator element. And we already talked about barrier sequences. Actually, barrier sequences work in exactly the same way. They're proteins that associate with them and will block the spread of some kind of chromatin, euchromatin, heterochromatin, etc. So it's binding of these insulator binding proteins that will block the activity of this regulatory binding sequence, again, usually an enhancer binding proteins, from interacting in the wrong place. And it turns out these are present all throughout different chromosomes. So these are Drosophila polytene chromosomes here. And lots of these insulator elements, which are the ones in green, turn out to be between these different bands. And if you were to look at genes that are being expressed, the puffs that happen are going to be between each of these insulator sequences. So again, insulator sequences, insulator binding proteins will associate with them, block the activity of an enhancer that's between those insulators from acting on anything other than what's right between them. Okay, we're going to talk about examples of all of these things in the next couple of lectures. But first, because it's that time of day, we'll have another clicker question. Which of the following is the most likely thing that insulators block? DNA bending, formation of heterochromatin, formation of euchromatin, activity enhancer binding proteins, activity of the mediator complex. Still aiming for 100%, right? Ten, 5. Okay, we're not at 100%, so let's try to get to 100%. Talk to your neighbors. Any of them. All of the above. I didn't put F on there, so there's no clicker button for F. Okay, we ready to go again? Yes, no? Go. Again, feel free to continue to discuss here. Push each other's buttons. 10, 5, okay. Nobody likes DNA bending, and in fact, they, if anything, stimulate DNA bending. Formation of heterochromatin and euchromatin, this is actually literally the formation of these, so it has actually nothing to do with formation, one or the other. Activity of the mediator should be there anyway, so we're only left with the enhancer binding proteins. And so it's the activity of the enhancer binding proteins, and it's blocking that they're going to the wrong place. So that's D is correct. Okay, so I've been beating around the bush a lot about chromatin so far. So here I have a gene activator protein that's bound to your DNA, and a Tata box which is near it but also connected with this DNA and completely bound up with nucleosomes. So there's no way that you can actually get the holoenzyme, or even for that matter, all of the general transcription factors associated with this Tata box. So how can you get this gene activator protein to actually activate this gene? There are lots of different ways to do that. The most straightforward is a histone modifying enzyme that your gene activator protein is going to interact with. So this would be your classic histone acetyltransferase, and it turns out that lots of co-activator proteins actually are histone acetyltransferases. And so they will bind to your gene activator protein, modify the histones, and in modifying the histones, allow other proteins to bind here that will release this Tata box from this promoter. You also can have histone chaperones, which will exchange nucleosomes in regions where you have now genes that you want to express, again, due to interactions with this gene activator protein. Sometimes those histone chaperones will put in specially modified nucleosomes, and we did talk really briefly about some of those specially modified nucleosomes, actually alternative histones, which are associated with transcription in some parts of the genome. And sometimes these histone chaperones will just get rid of all the histones at once, and you can have access to the Tata box from there. Or you bring in a chromatin remodeling complex, again, associated with your gene activator protein, and these remodeled nucleosomes pushing the nucleosomes forward and back relative to where you need to have the general transcription factors bind, which will now allow you to have interactions there. So I just wanted to finish up with two last slides here. These are a couple of different kinds of chromatin remodeling complexes. The names here are not really critical, but you have DNA binding sites for specific proteins that can either get put together here, and this one actually shuts down the activity of some genes, assembling nucleosomes, again, chromatin remodeling. This is blocked. You can't have any kind of activity here, so the activator can't bind to DNA. However, if you have a different remodeling complex, also again, through the activity of chromatin remodeling, again, always in the presence of ATP hydrolysis, that can allow the activator to actually bind. Different kinds of chromatin remodeling complexes can be this SWI/SNF family, which literally move the nucleosomes around or get rid of them completely, or you can insert a completely modified histone, like this histone H2AZ, which is important for getting your transcriptional activation. So actually, we'll stop here, and I'll start with this slide next time, which gives a bit of an overview on how these transcriptional activators are actually working.